Greetings, I'm Jack. This is a presentation about a book chapter from Marshner's Mineral Nutrition of Higher Plants, 3rd edition 2012, entitled Rhizosphere Biology. The rhizosphere, or otherwise known as the root zone of influence, is a narrow portion of soil about a few millimeters wide. It is inhabited by a unique population of microorganisms, often influenced by chemical released by the roots. The rhizosphere is composed of three zones. The endorhizosphere, found in the inner section of plant roots, is composed of the cortex, endodermis. Microorganisms and cations often occupy the space in between cells. Next, we have the rhizoplane, the zone directly adjacent to roots. It includes the root epidermis and mucilage. Finally, we have the ectorhizosphere, which is the area which extends from the rhizoplane and into the bulk soil. The rhizosphere is defined by a gradient of chemical, biological, and physical properties. The figure at the bottom represents different plant species and their unique rhizosphere. Plant roots release secretions, which are compounds actively released by the roots, and diffusate, which are compounds passively released due to osmotic differences between the roots and soil solution. These root exudates are a wide range of compounds that affect the nearby soil microbiome and initiate plant-microbe interaction. Root exudates contain different types of compounds. There are two categories for these compounds: the high molecular weight compounds, which are the root mucilage and cellulose. These are compounds not easily used by soil microbes. The next one is the low molecular weight compounds, which are easily metabolized by microorganisms. These are sugars, organic acids, phenolics, and secondary metabolites. On the table, we find different types of compounds found in wheat root exudates. We see different types of sugars, amino acids, organic acids, nucleotides, and secondary metabolites and enzymes. There are several functions of root exudates, one of which is to act as a signal for soil microbe recognition. Root exudates contain certain metabolites that attract and screen for specific soil microbes. An example of which is when root exudes flavonoids to attract rhizobia, and quercetin and strigolactone to stimulate the germination of AM fungal spores. During low phosphate conditions, strigolactones are produced in the roots. When detected by AM fungi, spore germination and hypho branching is triggered. MYC factors, or MYC factors, produced by the fungi gets recognized by the root cells, allowing colonization. Other functions include suppression of soil-borne pathogens, the stimulation of the production of phytohormones in microorganisms, like in rhizobia, where bacteria produce nod factors that stimulate auxin-cytokinin signaling pathways and stimulate growth of root nodules. They also contain precursors for phytohormone production, like tryptophan for IAA synthesis and L-methionine for ethylene production. There are several factors that affect microbial colonization of roots. These are the area of exudation or root zones, plant species and plant age, the distance covered by the exudates, and the type of exudates produced by the plant. In the table, we see a comparison of the attracted zoospores between avocado and mandarin oranges. In avocado, the number of zoospores decreases as you go further away from the root tip. Meanwhile, in mandarin oranges show a different trend where there are certain areas with higher and certain areas with lower accumulations of zoospores. Rhizosphere microorganisms are microbes that contribute to the plant nutrient turnover. They do this through mineralization of organic matter and through the release and transformation of inorganic minerals via solubilization, chelation, and oxidation reduction reaction. There are two common types of rhizosphere microorganisms, the infixing bacteria, and the mycorrhizal fungi. In this presentation, our focus will be on mycorrhizal fungi. Rhizosphere microbes greatly influence several aspects of plant morphology and nutrition. They affect growth and overall physiology, as well as nutrient availability and uptake. Rhizosphere microbes help with cycling of nitrogen by ammonification, nitrification, and mineralization. They also cycle phosphorus for plant uptake, utilization of inorganic phosphorus, and mineralization of organic phosphorus. Other microbes can release organic acids, anions, or siderophores that chelate and mobilize iron. They are also responsible for reduction and oxidation of manganese. Mycorrhiza or mycorrhizal fungi are fungi that form intimate interactions with plant roots. Majority of the plants on Earth form symbiotic relationships with them. Mycorrhizal associations can either be mutualistic, neutral, or parasitic in nature. There are two major groups of mycorrhizal fungi. The endomycorrhizas are fungi that penetrate through cell walls and anchor in the root cells. This enables the plant and fungi to be in direct contact with each other, while ectomycorrhizas are fungi that form interactions without penetration, usually interacting with the plant through intercellular spaces. There are three types of endomycorrhiza. The arbuscular mycorrhiza are the most abundant endomycorrhizas on Earth. They form arbuscules, internal hyphae, and external mycelium. There are two types. The arum types form arbuscules, and the paris types form hypocoels within cortical cells. Another type of endomycorrhiza is the ericoid mycorrhiza, which forms hypocoels within root cells. The next step is the orchid mycorrhiza, which forms symbiotic relationships with orchids. Orchid embryos have almost non-existent endosperms. These mycorrhizas provide an alternative source of carbon. Ectomycorrhizas are basidiomycetes or ascomycetes that produce hyphal strands or rhizomorphs. They form a mantle of fungal sheath that surrounds the roots. They also produce heartig nets, which are mycelial networks that form when hyphae penetrate the intercellular spaces of the root cortex. Root colonization by mycorrhiza are affected by the presence of spores, colonized root residue, and pre-existing mycelia. They are also affected by root exudates, the soil CO2 concentration, and the presence of helper bacteria. Genotype also affects mycorrhizal root colonization. AM colonization vary between different cowpea cultivars. Soil phosphorus limits colonization. In subterranean clovers, percent root length colonized by mycorrhiza increases as soil phosphorus reaches a certain point, and then decreases if soil P increases beyond this point. Respiration is 20 to 30% higher compared to non-mycorrhizal plants. In the table, different AM fungi affect colony units of bacteria, N2 fixers, and actinomycetes in Panicum maximum. P maximum with infected AM fungi generally had an increase in rhizosphere population. Nutrient mobility is affected by the presence of AM fungi. In the figure below, plant dry weight in both citronella and star oranges infected with AM fungi were both higher than plants without AM fungi, but growth eventually slows down as P supply in the soil increases above 200 milligrams per kilogram soil. The species of AM fungi also affects growth of plants. A higher shoot dry weight was observed in plants that form relationship with Aclospora as compared to Scutellospora and non-mycorrhizal interactions. In the first table, Plants with AM fungi at low P supply have higher dry weight and a higher number of nodule formation. It also has a higher nitrogenase activity compared to plants without mycorrhizal interactions. Meanwhile, the presence of nitrogen also affects nutrient uptake and mycorrhizal interactions. A higher dry weight and N content was observed in mycorrhiza-infected soybean and corn. This happened when nitrogen source was added. Mycorrhizas have been known to increase plants' tolerance to heavy metal exposure. They do this through several ways. They bind excreted ligands to their external mycelia. They sequester metals by binding them to the fungal cell wall. And they increase metal flux. The fungal cytoplasm also allows metals to bind to the melatonin or glutathione. They also sequester heavy metals to vacuoles. Mycorrhizas are also known to produce phytohormones or trigger their production in plants. ECM fungi can produce their own IAA to cause changes in infected root tissue. The pictures below show several roots that are infected by ECM fungi. Generally, infected roots are shorter and highly branched. They call this process bifurcation. The first root on the right is caused by an unknown mycorrhizal fungi infecting pine. The roots on the far left corner, which are black, are caused by an ECM fungi Xenococcum in spurs. How the fungi affect plant root morphology is highly dependent on the ECM species. Mycorrhizal fungi also have a role in suppression of plant pathogens. In the table on the right, we see the effect of AM fungi on replant disease in grapes, caused by Pseudomonas. Population of Pseudomonas is high in diseased plants without AM fungi. When AM fungi is present, the population of Pseudomonas in diseased plants is low. The next table shows seedling mortality when Pinus resinosa seedlings are inoculated with Fusarium oxysporum and an ECM fungi. Inoculation of sick plants with ECM fungi decreases seedling mortality. Mycorrhizal fungi are often used and developed for horticultural use, reforestation, and rehabilitation. It is also used in reclamation of mined lands. Reforestation success of hardwood seedlings drastically increase when inoculated with mycorrhizal fungi, as seen in the table below. The figure on the left shows the yield of marketable fruits of green peppers. The addition of mycorrhizal fungi in compost improved yield. This concludes my presentation on rhizosphere biology. Thank you.